Welcome to our stop motion video talking about different forms of cell signaling and their consequences. Within the cell cycle, there is a key factor of cellular division that must be understood when we talk about a cellular response. In interphase, during the S phase, the DNA replicates within a cell to ensure that there are a proper amount of chromatids that separate into two different cells. This occurs first by helicase unzipping the DNA from 5' to 3'. The primase then acts as a primer or a template for the DNA polymerase to come through and add the proper strands to create the proper pairs. After DNA polymerase does its job, ligase comes in to make sure that there are no nooks, crannies, or errors that could potentially cause a genetic mutation. And now you have properly replicated DNA. One of the main responses that cell signaling causes is mitosis. After the cell spends most of its life in interphase, it's ready to replicate. First, we enter prophase, where the nuclear envelope dissolves and the spindle fibers begin to form. In metaphase, the chromatids form along the equator of the cell, and the spindle fibers attach onto the chromatids, ready to pull them apart. During anaphase, the spindle fibers pull the chromatids apart at the centromere, and we now have one pair of chromatids on one side and one pair of chromatids on the other. We then enter telophase, where a cleavage furrow starts to develop, and the cytoplasm starts to pinch right at the equator. The nuclear envelope starts to reform, and the spindle fibers begin to disintegrate. We then have cytokinesis, where the cell splits entirely. Our first form of cell signaling will be GPCR, or G protein-coupled receptor signaling. When a ligand binds to a receptor on the cell membrane, it causes a G protein to be activated with GTP. GTP knocks off the GDP and causes the G protein to bind to an enzyme. When this occurs, the enzyme is activated and creates cyclic AMP, which is the second messenger. Cyclic AMP comes from the breakdown of ATP into ADP and a diphosphate bond. This cyclic AMP acts as a second messenger, where it relays the signal down a phosphorylation cascade of kinases. A phosphate moves down the kinases until it reaches the final protein. This protein is then activated, which initiates a cellular response. Our second form of cell signaling that we'll be talking about is ion-gated channel cell signaling. First, a ligand binds to the channel and activates it. This causes a conformational change where it opens and allows ions to pass through. The ions move across the membrane according to their electrochemical gradients. Then, the ion movement causes a change in the membrane potential, which initiates a cellular response. For example, action potential or muscle contraction. After the ligand is removed, the channel closes and it goes back to its dormant state. The third version of cell signaling that we will be talking about is receptor tyrosine kinase cell signaling. First, a ligand binds to an extracellular domain of the RTK. Then, as the ligand is binding up to them, the RTK monomers dimerize or pair up. Autophosphorylation occurs when the intracellular tyrosine kinases of the dimerized RTKs phosphorylate each other on specific tyrosine residues. Then, phosphorylated tyrosines act as docking sites for intracellular signaling proteins. Activated signaling proteins trigger a downstream pathway that regulates cell functions such as growth or survival. These signaling pathways lead to changes in gene expression, cell cycle progression, metabolism, or survival. The signal is then terminated through dephosphorylation or endocytosis of the receptor, then halting the pathway.